Hi guys, my name is Pang. Welcome back to my channel. Today I'm going to be showing you my home tissue culture lab, which essentially now is my full-time job, and show you around how I operate my small house lab and a little bit of my workflow. I start off with my media. The media has been sterilized by the autoclave. I keep them inside the container, and as you can see I have two types, the first being the ones in BA and the second without hormones. And now I have my forceps, magnetic bars, scalpels, papers, and these are all sterilized by the autoclave. I like to keep my equipment organized by compartmentalizing everything, as you can see. I have pre-treated my laminar flow hood with the UV lamp for 30 minutes. Then I wipe out the surface of a table with 75% isopropyl alcohol. I now turn on my glass bead sterilizer and wait until it gets to the recommended temperature. I used to use a Bunsen burner for in my lab, and I quickly switched to a glass bead sterilizer after an incident where I almost burned down my own house. That's a story for another day. Next, I open two packages of surgical blades, and I need to be very cautious with handling the blades because they are super sharp. And I always have an extra set of scalpels and forceps so that I can switch the tools around and wait for the other one to cool down. And always, while using the tools, make sure that they are not too hot because they will damage and potentially kill the plants. And again, be very careful when inserting the blade. It shouldn't require much force, and with the right angle, it should slip into the lock with ease. By now, some of you might wonder why I have a timer in my flow hood, and that's because I always forget how long I've put my tools in the hot beads sterilizer. Leaving the tools too short will result in the tools not being sterilized, and too long, then the blades will lose its sharpness pretty quickly. These jars were sprayed down with 75 isopropyl alcohol, then inserted into the hood. I carefully use my forceps to handle the sterilized paper and place it onto my cutting tray. Side notes, always try to avoid using your hands to unnecessarily touch anything inside the hood. The first plant I will be working on today is a variegated caramel marble. I carefully chop the specimens and put them into different jars. As you can see, I have prepared two colors of media. The clear one is just MS without hormones, and the yellow jar containing 1BA. I inserted food coloring to give my agar some color, so that I can use it as a way to identify the media type without having to write or attach a label. When transferring the plants into new media, avoid any surface touching of the forceps onto the media to avoid contaminations. As you can see that some of the plant has very high variegation and some has none. I suggest if you want to keep the plant nicely variegated, always choose wisely the plants you want to subculture. Think of it as a copy. The nice ones you want to keep copy of, and the bad ones, it's better if you discard them right away so that you don't have to waste time and resources on it. This video is just a brief insight into my lab workflow. There are much more details that I cannot compile into this one video, so be sure to ask questions if you're interested in anything, or just subscribe for a more in-depth video in the future. Next up is the variegated Billietaie. This plant specifically is a little hard to subculture. The plant divides very slowly. Whenever subculturing, always make sure to remove all the dead leaves, since they can potentially become a source of contamination in the future. Be sure to wait until the scalpel is no longer heated before proceeding to cutting any kind of plants, because it can potentially damage your explant. Here, as you can see, I carefully remove the old leaves, the yellow ones, and keep the nicely variegated plants. Another tip of subculturing a variegated plant is never to use high amounts of hormones, because they can result in fully variegated plants or all green plants. Each species of plants react differently to types and amounts of hormones you put into the media, so you really have to experience and play around with it. For variegated philodendrons, I will make a video catering especially for it in the future episodes. These jars are not 100% airtight. Therefore, I use a plastic food wrap to make sure that the container is 100% airtight to avoid any potential contamination in the future. I then use a permanent marker to put a label onto the jar. I like to write the names and the dates so that I can keep them organized. I then remove all the used tools out of the flow hood and store the scalpels and forceps into the used box. I will later detach the blades and clean them up, and then re-sterilize it for the next usage. And these are some of the final products. And as you can see here, these are some nicely variegated caramel marbles. And this is a Billietaie. Then I put it under my grow light. And as you can see, it's a bit cramped up in here, but I don't have any more space yet. So I'm working with my tiny space here. And I also acclimatized some. So these are the outcomes. That's it for today, guys. Thank you for watching till the end. I will post more videos for people who are interested in low budget tissue culturing, because I also started from a very cheap DIY stuff, I understand. So be sure to subscribe for new tips and tricks videos in the future. Bye!